The broadcast is now starting. All attendees are in listen-only mode.
Good evening, everybody. Welcome to our class, Mechanism of Genetic Exchange. Today, we're going to be discussing more about genetic exchange. We want to see what we meant by genetic exchange. Well, let's kind of start by looking at what these French scientists, who also double as a Nobel laureate winner, Jacques Monod. Jacques Monod in 1954 uttered these famous statements. What is true in E. coli is also true in the elephants. And what does he mean by this? It was actually suggesting that the biochemistry of life, whether it be in small bacteria, can also be shared by the elephants. Since that famous statement by Monod, we have learned a great deal about the mechanism of gene regulation, gene expression, and replication in living cells. We know fully well that cells use DNA for information storage. They also share the same genetic codes and use similar mechanisms to replicate as well as to express it. Although many aspects of genetics are universally shared, there are variations that exist among these genetic systems. We now know that within the shared overall theme of the genetic mechanisms, that there are significant differences among the three domains of life, which are the eukarya, archaea, and the bacteria. Additionally, we also know that viruses, cellular parasites, not to talk of the living cells, also show dramatic variations in their genetic materials and their replication and gene expression processes. So some of these differences will allow us to look at how to engineer chemical tools such as antibiotics and antiviral drugs that will specifically inhibit the reproduction of pathogens, and yet will not be harmful to the host. So let's just quickly start. That's just an introduction to genetic exchange. What do we understand by the word genes? Genes. Gene is composed of DNA that is read or transcribed. Is read or transcribed during the process of transcription. One major type of RNA molecule, which we refer to as the messenger RNA, provides the information for ribosome in order to produce protein through a process called translation. So we've mentioned three processes here. With the gene that is transcribed to an messenger RNA. This messenger RNA, we bring about the translation of what is transcribed to form an expression, gene expression. So kind of look at what we have here. This is overview of replication. If you're looking at the screen, this is a DNA molecule. This DNA molecule can be replicated. The DNA molecule will be duplicated, then it will lead to transcription of that DNA as it is copied onto an RNA. This is a single-stranded RNA. The DNA is a double-stranded. Why this is a single-stranded RNA? This RNA is now translated to produce proteins. But normally DNA will first be replicated before we now talk about transcription and translation. Now, DNA replication. The DNA replication process is semi-conservative, which implies that we can take it from left to right, semi-conservative. And this results in two DNA molecules. Okay, now this is a DNA. This is a makeup of a DNA, double helix DNA. This is sugar phosphates. You can see this side here. This is a 5' sugar phosphates. But when you take a look at the other side, it's a 3' sugar phosphates. So these 3' and 5' we refer to it as a semi-conservative. So the strands of a DNA are anti-parallel to each other in their orientation. 3' direction, and its complement is oriented in the 3' to 5' direction. So it's a semi-conservative process, which results in two DNA molecules, each having one parental strand of DNA and one newly synthesized strands. So if this is a replication, this is what we're likely going to see. This replication of the DNA. Now, in bacteria, the initiation of replication will occur at the origin of replication, where supercoiled DNA is unwound. It will be unwound, split open. It will be unwound open like this. This is the unwinding of the DNA molecule. So we'll take a fragment here. See, the DNA will be unwound and or split open. So unwind by this is a DNA polymerase or DNA gyrase made single-stranded by helicase and bound by single-stranded binding protein to maintain a single-stranded state. So this DNA is being unwound, being open, to be open like this, to be open. Now, it will form a new strand. This is a new strand. That is the RNA transcription. Now, during the process of transcription, the information encoded within the DNA sequence of one or more genes is transcribed into a strand of RNA. This is our DNA being unwound, has been opened. Now, this is the transcription of a new strand using the DNA polymerase or the gyrase to produce an RNA, as you can see. And this is the 3', the 5'. The resulting single-strand RNA molecule is composed of ribonucleotides containing the base adenine, cytosine, guanine, and uracil. acts as the mobile molecular copy of the original sequence. Now, this is what we are seeing. They will copy shoulder. This is the 5' copy to form all these type. This is genetic codes. We'll get there later. So, but this is the unwinding of the form. Now, that takes us to, after transcription, let's look at protein synthesis. Protein synthesis. Now, in protein synthesis, the synthesis of protein consumes more of a cell's energy than any other metabolic process. In turn, proteins account for more mass than any other macromolecules of living organisms because of its body building structure. Another macromolecule of living organisms. They perform virtually every function of a cell, serving as both functional, for example, enzymes, and structural elements. The process of translation of protein synthesis, the second part of gene expression, involves the decoding by a ribosome of a messenger RNA message into a polypeptide product. So this now what will actually happen is that when it leaves the gene, we now have to, this single-stranded will have to leave, and after that's left, it will now be translated by the ribosomal RNA. So let's try and understand what we are saying. The gene, which is the materials that carry genetic information, is basically on the DNA. The DNA are made up of base pairs. Base pairs, like we can see here. This is T here stands for thymine, cytosine, guanine, adenine, guanine. These are base pair. Base pair. Now, this base pair will be transcribed. The DNA, this DNA will unwind at a point here. It will unwind and open. And after opening, the DNA polymerase will travel around it and start to read these strands, or read the strand in order to transcribe what is here. So let's take, for example, I have AGCTC. Now, A will pair with T, G will pair with C. So whenever you see A, the next base pair will be T. When you see G, the base complementary base will be C. If you see C, the complementary base pair will be G. And if you see T, the complementary base pair will be A. So after the unwinding of the DNA molecules, the polymerase will read it and transcribe the information onto an RNA. That RNA is what we refer to as a messenger RNA. Now, it will transcribe it in a semi-conservative pattern, whereby it starts from the 3' to the 5'. If it is a 5' to the 3', it will be 3' to 5'. So they are anti-parallel to each other. So the information will now be transcribed onto an mRNA, as a messenger RNA. Now this messenger RNA we then have to move away into the cytoplasm, where it will now find itself in the ribosomes. In the ribosomes, that's where translation of the messenger RNA will occur. So the messenger RNA will now have to be translated into forming different amino acids, and the joining of these amino acids to several amino acids to form a polypeptide, which eventually give rise to the formation of protein. So we've seen the third process. The third process is the replication of the DNA. The DNA needs to be replicated. That's the first thing. You need to replicate the DNA needs to be replicated. And after the DNA has been replicated, the DNA will also be transcribed. There will be a the DNA will be transcribed onto the messenger RNA. Then after being transcribed to the messenger RNA takes the transcribed information, the coded information into the ribosomes, where these codes that are on the messenger RNA will now be read and translated to form proteins. So now protein synthesis, which is translation, the translation of protein, like we said, will consume a lot of things. Now, in genetic information, during this genetic information, there are instances where there will be error as a result of translation of these coded message to form protein. When such happens, it will lead to what we refer to as a mutation. A mutation. Now, what is a mutation? A mutation is a heritable change in the DNA sequence of an organism. The resulting organisms, which we call a mutant, may have a recognizable change in its phenotype compared to the wild type. So the wild type are the real one. Now, if there is any change during this process of transcribing, translating, it will lead to the formation of a new strand or a new species, which is a kind of changes, there can be changes in the phenotype. Now, which is the phenotype most commonly observed in nature? A change in the DNA sequence is usually confirmed on the messenger RNA through transcriptions and may lead to an altered amino acid sequence in a protein during translation. So what that simply is telling us is that if, for example, during transcription there is a mistake, something happened. So instead of we having a T, we have A here and we have T here, and there's a mistake. This one is omitted. The T is omitted. We'll now jump from C, G, A, G. C, G, A, G, we amount to something else. Will give us something else, because these three amino acid base we code for a particular, these three nucleotide base pair we code for a certain amino acid. And when the T has been omitted, it will form a different form of species. And that species is what we call mutants. Mutants. Now, we know fully well that most organisms, especially at the cellular stage, cannot carry out sexual activities where gene is exchanged from one sex to the other. So question is, how are asexual prokaryotes able to carry out genetic diversity? Now, how do organisms whose dominant reproductive mode is asexual able to create great diversity, especially in prokaryotes? And that's where the introduction of genetic material from one organism to another organism within the same generations is key to understand the transmission of gene from one organism to another. We're going to learn how the microorganisms are able to transfer their genetic information right from one organism to the other. We're going to learn how bacteria are able to do that. We're going to learn how the virus are able to do that, since they don't have sexual organs. Unlike matured organisms, higher organisms like plants and animals, they have reproductive organs which can help them to transfer genetic information from one sex to the other. But these ones are actually asexual organisms. We're going to learn the different methods by which they move their genetic information to another. Now, this is a direct transcription of what we have been talking about. Is a kind of, this is the RNA polymerase. RNA polymerase will unwind this, and when it's unwinded, they start to replicate and produce it. So in other words, like I did mention, we have a lot to learn about the genetic exchange of different organisms. We just want to understand the basis. By next week, Monday, God's willing, we'll take a look at the different mechanisms by which bacteria and viruses are able to transfer their genetic materials from one organisms to the other. Genomic DNA contains both structural genes, which are encoded products that serve as cellular structures or enzymes, and the regulatory gene, which encode products that regulate gene expressions. So the expression of a gene is highly regulated process, whereas regulating gene expressions in multicellular organisms allow for cellular differentiation. In single-cell organisms like bacteria, viruses, prokaryotes, it ensures that the cell resources are not wasted, so making proteins that the cell does not need at that time. So that's what gene regulation theory states, that is to allow materials not to be wasted. So these are things. So like we were saying is that in genetic exchange, the means of genetic exchange is the DNA. The DNA is the genetic material that contains all the information about a living organism. All the informations, the characteristics of that living organism is coded onto a DNA molecule. Now, the DNA molecule, we have to be replicated. So we need to reproduce the DNA. And after reproducing it, it goes through a series of chemical processes. series of chemical processes. The replication method go through series of chemical processes where a DNA polymerase, an enzyme's DNA polymerase will be utilized. So in this aspect, if there is going to be a production of a protein, what will happen is that the DNA will be unwound, will be open. Now, when the DNA is unwound, open up, unzip, will be unzip, it will form a template. We have two templates, one for the parents and one that will form the new synthesized daughter cells. So it is these new daughter cells that will come out. So that fragments that has been copied will be meant for the use of a particular protein that is needed. So that's replicated DNA will now have to be transcribed. Don't forget that the DNA is found in the nucleus of a cell. So this particular DNA that has been replicated or reproduced will now have to be transcribed onto an RNA. That particular RNA is called messenger RNA. Now, the messenger RNA will copy the replicated protein anti-parallel. That means when it's forming it's formed through a semi-conservative process, whereby it is from 5' to 3'. That's how it will reproduce itself onto the RNA. And when it reproduces itself onto the RNA, we talk about transcribing. That's the DNA has been transcribed. So that's where transcription comes. And the DNA has been transcribed onto a messenger RNA. Now, this messenger RNA will now have to leave the nucleus of the cells and travel out onto the ribosomes. In the ribosomes, there are two types of ribosomes. There are two types of ribosomes. There are two types of RNA in the ribosome, two types of RNA in the ribosomes. One is what we refer to as a transfer RNA, and that's tRNA, small t. Then the other one is RNA, that's a small r, that's ribosomal RNA. Now, the ribosomal RNA will have to translate the code on the messenger RNA. Now, they are able to do that by reading the codes on the RNA. Let's take, for example, this is what we find on the RNA: UUU, UUC, UUA, UUG. Each of these base pair or these codons, call them codons, simply means a particular amino acid. So for example, now UUU is phenylalanine. UUC can also be translated as phenylalanine. Now, if you have UUA, leucine, or you have UUG, leucine. You have CUA, leucine, CUG, leucine. Now, all these amount to a particular type of amino acids. Now, the ribosomal RNA will read it. After reading it, the transfer RNA will now go to the pool of amino acids that are present in the body cells and bring the particular amino acid and attach it to the ribosome. It will continue in that fashion by joining joining amino acid together through a peptide linkage, until finally it will get to a stop codons. There are stop codons here, so if we take a look at the distance, probably we'll see. Methionine is a stop codon, which tells the rRNA, ribosomal RNA, to stop producing. So when it gets to the stop codons, when it picks the stop codon, it will signify the end of such protein formation, what is now referred to as the translation of protein. Now, this protein will now be translated. These translated proteins, we have to undergo foldings. Remember, we've mentioned before that protein have different structures. During the production of the protein will be having its primary structure because it will be linear structure while they are binding together. Subsequently, through the interactions of different bonds on the protein, it will lead to the formation of quaternary protein, which will now be used for the purpose at which the protein was made in the first instance. Now, to prevent mutations, this process is highly proofread. To prevent mutation, the process will be screened properly to avoid any mistakes that might occur during the process of translating it to the production of proteins. But at times, even with the best method of proofreading, there are instances where there are mutations. So mutation can occur occasionally, but that is very rare when it comes to replication. So it is very important for us to understand this, because these are things we're going to learn subsequently. Now, the DNA molecule are they are double helix. Folded in the chromosomes, but sometimes they are in the form of a plasmid. A plasmid is a circular molecules of DNA, a plasmid, which you find in some bacteria, which help them to be resistant, disease resistant. They are very resistant to drug, and it helps them to have virulent factors which can make them to cause diseases. So we're going to be discussing these in our subsequent class to see how this gene will be transcribed or be translated, be exchanged. The particular organisms can carry out exchange. In higher organisms, like we mentioned, there's what we call crossover. The mom, the dad, they undergo sexual relationship, whereby the sperm, which is the carrier of genetic information, will fuse with the egg, thereby allowing the genes to cross over to form a single genome. But in prokaryotes, like bacteria and viruses, there's no sexual activities occurring there. So it has to find different means by which this material will have to be exchanged between two different organisms, two bacteria, one exchanging material to the other. We're going to look at the different, but the thing is that we want to understand the process of genetics, the mechanism of genetics. So today, what we have learned today is simply the mechanism of microbial genetics. Tomorrow, we're going to look at, Monday, God's willing, we're going to be discussing the mechanism of microbial, of genetic exchange inside microbes. That's what we've been discussing. So today we'll learn the mechanism of microbial genetics. In our subsequent class, we're going to learn the exchange of these medicines, how it occurs. So the process is what we're going to learn. So we're going to stop the class today. I believe we have understood the microbial genetics. We understand what the DNA is all about. We have also understand the RNA. We should know fully what the difference between a DNA and RNA is. One, the DNA is a double-stranded molecule, while the RNA is a single-stranded molecule. The DNA has a deoxyribonucleic sugar, a deoxyribose sugar, while the RNA have a ribose sugar. That's a ribose sugar. A DNA has thymine as one of its nitrogen base, while for your RNA, it is uracil. So the thymine will not be there. All you have is uracil. So these are basically the difference between an RNA and the DNA. So the DNA is where the genetic information is actually stored. The genetic information is stored in the DNA, while the RNA, the genetic information from the DNA will now be transcribed onto the RNA. There are three types of RNA. Like the DNA that is one, there are three types of RNA. You have the mRNA, you have the rRNA, and you have the tRNA. The mRNA stands for the messenger RNA. The rRNA is the ribosomal RNA, while the tRNA is the transfer RNA. So all these three RNA together with the DNA will work together to give rise to the synthesis of protein through a process known as translation. Now, the cell is well organized to ensure that there is no mistake in the process. But even at that, there will be a rare occasion, a kind of change in DNA sequence. And when there is change in DNA sequence, we say there is mutation. So the organisms has become immune. So these are some things we need to just understand. Like I said, subsequently, we'll now be looking at how these material are now being exchanged between two different organisms. So if we have any question, feel free to send it to my email. I'll look at them and send you a reply. Thank you very much and good night.